we all use different types of gonadotropin ampoules, and these ampoules are either urinary HMG, urinary FSH, highly purified HMG, highly purified FSH, urinary HCG, rec HCG, rec FSH, or rec LH. Most of the urinary products are composed of FSH, LH, and HCG. And as you can see, the alpha chain of all of them has 2 N-linked glycosylation sites, whereas the beta chain is different. In LH, it has 1 glycosylation site. In HCG, it has 6 glycosylation sites. And these 6 glycosylation sites resulted in HCG having long time of duration of action. And this is why ampoules which contain HCG can accumulate HCG in the circulation, and this could be dangerous unless we took care of the dose that we are giving. During purification of HMG from urine, LH is lost and is replaced by an LH activity that is composed now mainly of HCG and a little amount of LH. The HCG content or concentration of highly purified products is more than the HCG concentration of the gonadotropins, human menopausal gonadotropins. And also the 2 highly purified gonadotropins differ in their content of HCG and the type of HCG that they have. FSH in all urinary products is an acidic FSH, which means that it has cyaric acid on the carbohydrate moieties, and this gives the product a specific characteristic. It is long-acting and it doesn't bind strongly to the receptor, so it gives a mild stimulation. Whereas in rec FSH, the FSH is basic, which means that it has sulfonic acid on the carbohydrate moieties, and this will result in a compound which is potent. It binds strongly to the receptor, but it is short-acting. So again, FSH in all products has alpha and beta chain, and the alpha chain has 2 N-glycosylated carbohydrates, and the beta chain has 2 N-glycosylated carbohydrates. If the carbohydrates are linked to cyaric acid, mainly the compound is acidic, and if it is acidic, the estradiol resulting is low, cyaric acid content is high, receptor affinity is low, and the half-life is long. Whereas if the compound is basic, that it contains sulfonic acid like in rec FSH, it has high E2 resulting from it, and the receptor affinity is high, and the half-life of the compound is short. Another important thing is that physiology gives us lessons. Early in the follicular phase, follicles which are less than 10 millimeter have no LH receptors on their granulosa cells. Whereas after 10 or 12 millimeter, they start having LH receptor on their granulosa cells. These follicles, the small follicles, are recruited in the natural cycle by acidic FSH, the one that contains a lot of cyaric acid, like the urinary products. Whereas after the follicle reaches 12 millimeter, it is maintained in growth and function mainly by LH in the natural cycle. So if we want to simulate what is happening in the natural cycle in treatment, we should speak about selective action of gonadotropin in the natural cycle. So before 12 millimeter, there is no LH receptor on the granulosa, and the small follicles are recruited by acidic FSH. After 12 millimeter, LH receptor present on the granulosa cells, and small follicles are under the control of LH mainly, which allows the follicle to grow and estradiol to be secreted. Selective use of gonadotropin or personalized ovarian stimulation means that stimulation should differ when the follicles are small as compared when the follicles are larger than 12 millimeter. And this is the registered randomized control trial of EFRE multicenteric. And in this randomized control trial, we use two protocols. One of them is FSH followed by highly purified HMG, whereas the other is FSH and highly purified HMG followed by also highly purified Mg when the follicle exceed 12 millimeter. So this is our protocol. FSH is given only when follicles are small till day 6 or when follicles exceed 12 millimeter, and then 150 highly purified HMG is given. The other protocol, 150 FSH and 75 highly purified HMG till size 12, and then 150 highly purified HMG till the criteria of HCG are attained. The conventional protocol used to be FSH and highly purified HMG all over. And in the MEGASET study, the protocols were protocol 1, FSH till day 6, then individualized FSH doses, or highly purified HMG till day 6, then individualized highly purified HMG. So in the MEGASET studies, one protocol all through, there is no variation of the protocols or the compound between day 6 and the others. So in this small study, in this small presentation, I wanted to show that ampoules are different, that follicular genesis is under control that varies when the follicles are small as compared when they are large, and that selective gonadotropin could be a nice approach if we want to follow this. And the evidence, of course, has to be through randomized controlled trials. Thank you very much.